Yes, can you hear me?
Yes, I can hear you. And we're bringing up your slides and we will share them. And you're just going to need to tell us when to go forward. We're going to make it work.
Okay, so I'm trying to share my screen.
Nope, but we actually have yours and we can share your screen.
Okay, I can see it. So we can, okay, so I can start like this.
Yep, and then just tell us when to go to the next one.
Okay, so I will talk about the hemodynamic abnormality that initiates salt-induced hypertension in a rat model of primary aldosteronism. Next, please. Primary aldosteronism is a well-known cause of salt sensitivity and salt-dependent hypertension. Next. In a setting of high salt diet, increased level of aldosterone will increase blood pressure much more than a high salt alone. The aldosterone amplifies the effects of salt on blood pressure. Next. In a setting of low salt diet, aldosterone has little or no effect on blood pressure. It turns out that prevalence of primary aldosteronism is high. Primary aldosteronism may be present in 25% of hypertensive and 15% of normotensive, and it could contribute to nearly 50% of cases of salt sensitivity. Next, please. So this raises the question, how does aldosterone promote salt-dependent hypertension? Most people believe the traditional volume loading theory of Guyton and colleagues. Next, please. According to this theory, people with hyperaldosteronism are salt sensitive because increases in aldosterone increases the activity of epithelial sodium channels in renal tubules. This causes increased reabsorption of salt and water, increases in blood volume that raises blood pressure and initiates hypertension by increasing cardiac output. Next, please. According to this theory, people with normal aldosterone levels are salt resistant because when you give them salt, they suppress the aldosterone level and they rapidly excrete salt and have no increase in cardiac output. That is the theory most people believe. Next, please. But there is a problem. The theory is based on studies that did not include salt loading in controls with normal aldosterone levels. Surprisingly, no one has ever compared subjects with normal aldosterone levels to those with hyperaldosteronism with respect to salt-induced changes in cardiac output and vascular resistance. Next, please. So we designed the first control study of hemodynamic effects of salt loading in an animal model of primary aldosteronism. We used Sprague-Dawley rats that have been unilaterally nephrectomized and have implanted Doppler flow probes around the aorta to measure cardiac output continuously and telemetry arterial blood pressure probes to measure blood pressure. Next, please. Some of the animals were randomized to get a continuous infusion of aldosterone by osmotic mini pumps to raise the aldosterone levels about 4-fold. Next, please. The controls were getting infusion of vehicle solution. Next, please. And we studied them on both low salt intake and then during the transition to high salt diet. And next, please. And we monitor 24-hour hemodynamic parameters, mean arterial pressure, cardiac output, and systemic vascular resistance. Next, please. We tested the claim of Guyton, Hall, and others that in hyperaldosteronism, the salt-dependent hypertension is caused by primary increases in cardiac output, followed by secondary vasoconstriction. Next, please. Here are the results. There is no difference in mean arterial pressure between aldosterone-treated animals and controls when they are on low salt diet. The moment we switch them to high salt diet, within a day or two, blood pressure increases in both groups, but then levels off in control animals with mild hypertension. but the aldosterone-treated animals continue to have elevation of blood pressure, and thus aldosterone amplify the effects of salt on blood pressure and cause severe hypertension. Next, please. Here are the 24 hours averages of cardiac output. During the low-salt diet, there is no difference in cardiac output, but much to our surprise and against the guidance theory, when we added salt to the diet, cardiac output decreased in both groups, but much more in the aldosterone-treated animals. Next, please. So aldosterone-treated treatment causes salt-induced decreases in cardiac output. Next, please. Aldosterone-treated animals had greater vascular resistance. Vascular resistance thus accounts for initiations of hypertension. Next, please. This raises the question in hyperaldosteronism. What causes cardiac output to decrease during initiation of salt-induced increases in blood pressure? Next, please. As can be seen, aldosterone-treated animals had significantly greater stroke volume under low-salt diet, and when we introduce salt into diet, stroke volume increased even more. Next, please. In addition, we observe significantly decreased heart rate in aldosterone-treated animals, even on low-salt diet, when there is no difference in blood pressure. This difference is thus not simply due to baroreflex inhibition of sympathetic activity. When we added salt, then the blood pressure elevation magnified in aldosterone-treated animals, and there was further decrease in heart rate. So cardiac output decreased presumably as a consequence of increased systemic vascular resistance, decreases in heart rate, and potentially increases in afterload. Next, please. This raises another question. In hyperaldosteronism, which mechanisms promote increase in vascular resistance that initiate salt-dependent hypertension? Recent studies have shown that increased tissue sodium accumulation without a parallel increase in blood volume predisposes to hypertension. So we measure electrolyte in carcasses and in skin in these rats. And as can be seen in aldosterone-treated rats compared with vehicle-treated rats, switching from low-salt diet to a high-salt diet was associated with reduced tissue concentration of potassium prior to increases in mean arterial pressure and systemic vascular resistance, and decreases in heart rate and cardiac output that develop later during salt and aldosterone treatment. After initiation of hypertension, we can see secondary increase in tissue sodium in aldosterone-treated rats. Next, please. This slide shows that in aldosterone-treated rats compared with vehicle-treated control, switching from a low-salt diet to a high-salt diet was associated with reduced plasma concentration of potassium prior to initiation of salt-sensitive hypertension. Plasma potassium has a vasodilator function by stimulation membrane sodium potassium adenosine triphosphate activity, resulting in hyperpolarization and relaxation of vascular smooth muscle cell. Plasma potassium is also involved in nitric oxide production. Next slide, please. This slide shows that rats treated with aldosterone and salt showed no water retention when compared to vehicle-treated controls. Next slide, please. This slide summarizes our findings that aldosterone-induced hypokalemia precedes initiation of salt-sensitive increases in blood pressure and systemic vascular resistance, and decreases in cardiac output without water and salt retention. In addition, we analyze effects of aldosterone and salt treatment on expression of genes in the skin using RNA-seq method 2 days after salt loading aldosterone-treated rats, and compared to vehicle-treated rats, show differential expression of genes from biological process hyperosmotic response, while after 14 days of salt treatment, we observe differential expression of genes involved in biological processes, muscle development, and contraction. Next slide, please. The traditional volume loading theory of Guyton and colleagues holds that in hyperaldosteronism, the salt-dependent hypertension is caused by a primary increase in cardiac output, followed by secondary vasoconstriction. And this theory is not supported by our results. Next slide. Next, please. Instead, we believe in vasoconstriction theory. According to this theory, the salt-dependent hypertension is initiated by primary increases in vascular resistance, not increases in cardiac output. In fact, vascular resistance is driving decreases in cardiac output together with decreases in heart rate. Next. In primary aldosteronism, salt-dependent hypertension can be initiated by primary increases in systemic vascular resistance. Contrary to the traditional volume loading theory, increases in sodium-driven body water overload, followed by increases in cardiac output, are not necessary for initiation of salt-dependent hypertension in hyperaldosteronism. Next, please. Increases in systemic vascular resistance can initiate salt-dependent hypertension despite the occurrence of simultaneous decreases in cardiac output. Next, please. Aldosterone-induced hypokalemia may cause primary abnormal vascular resistance responses to salt by altering the balance in activity between vasoconstriction and vasodilator pathway. Next, please. It can be concluded that in primary aldosteronism, the hemodynamic mechanisms of hypertension initiation is better explained by the vasoconstriction theory than the traditional volume loading theory. Thank you for your attention.
Any questions? Oh, I thought Alan might have a question. You're gonna need to repeat it. Right.
Very nice study. The question is, how do you think about nervous system cultivation and how it looks for
Okay, Michael, we had a question. I'm going to try to repeat it. Can you say that again, Alan, and one at a time, and then I'll talk to him.
Yeah.
It's a very nice study from Alan Cowley, he's saying, and he had a question about, do you think the volume? Oh. So do you think the is it due to a CNS or vascular?
We think it is vascular, yes. Which the reason is that the gene expression profiles show something like involvement of genes that are responsible for development and contraction of smooth vascular cells.
So you think this is a primary thing that happens, or is it a secondary? That's sort of your question, Alan?
Actually, I think that the primary event that initiates salt-induced hypertension is actually at this moment. It's a rather hypothetical. It's the accumulation of sodium, and the main is actually the hypokalemia that we observe before the initiation of hypertension. We observe really profound decrease in the potassium concentration in tissue and plasma.
I can't see him.
Okay. Thank you.
Sorry, this is a little difficult because you can't hear us and we can't hear you. And so we're trying to repeat questions.
I can hear you quite well.
memory.
Yeah.
So thank you, Michael.
Okay, thank you too.
So do we have time for what, a 5-minute break, or if we run into lunch by 5 minutes, we okay? You want to do a 10-minute break or push through the next 2 talks and then do lunch? Break. Okay, we're going to break for 10 minutes.
Welcome back, everyone. The speaker is James M. Landler with a talk about the differences in microbiota that collaborate from access to the model of family from Thank you.
Oh, all right, hang on. I've got 2 different ones. Yeah, you can hold that. It's great to be back. It's been a long time since I've been to the RAT meetings. I really appreciate coming back and seeing a lot of old friends. It's been since probably Cold Spring Harbor since I've been to the RAT meeting. And so, and I'll talk a little bit about genetics. I threw some things in. I hope I don't go over. I know I'm keeping you all from lunch, and as well as I want to hear the talk after this a lot. So I appreciate that. So, all right, hang on. How do I go forward? All right, so I was reminded I do need to put this in perspective. We haven't had any cancer talks, so I certainly just want to bring you, back to the human. I think that's important to stipulate. And I think the big green wedge here is the wedge that we're kind of going after with model systems, right? There's this huge familial component that we don't really understand in humans in relationship to heritability of cancer. And certainly when we see that little blue wedge up there, knowing about hereditary cancer syndromes, it was really the APC min mouse combined with the human genome mapping that Vogelstein had done. And it was really that combined effort of human mapping and mouse models that happened to coalesce at the very same time to really identify that first tumor suppressor gene related to the familial adenomatous polyposis syndrome. And so that really kind of drove all of this idea of, can we resolve the rest of the genetics surrounding colon cancer and heritability? And certainly we know a lot about environmental factors like smoking and obesity, a lot of those things, but the relationship between those are just now being resolved. Certainly smoking and carcinogens, that's easy to find out. Obesity, it's a little bit more difficult to figure that out. And that relationship between obesity and dietary factors and colon cancer also is very interesting. And I'm going to try and address that in the context of the microbiome. So, let's see if I can. All right. So to give you a little context of why we're even studying the rat, I talked about the APC min mouse, but the difference in the kind of, we tried to sell the mouse as a great model of human colon cancer. Unfortunately, the vast majority of the tumors that arise in the APC min mouse are in the small intestine. So it doesn't look like the familial disease. And if it weren't for Michael Gould at the University of Wisconsin, I'd never be working in rat. And I'm sure you guys have talked about Michael in the past. And this is it's really an homage to the relationship that Michael wanted as many people involved in screening mutant animals in his screen at the time. That really led me to the rat. And so we did this screening of ENU mutagenesis in the rat, and we found a single mutation, this PERK mutation here, in the APC gene. And if you look and see where in the familial adenomatous polyposis families, where those mutations arise, they're really all throughout the gene. This is really the hotspot, the people that have most severe disease here in red. But the correlate position in human is pretty close. So we were pretty excited when we found that mutation, and we were hoping that it would have an interesting phenotype. So I threw this slide in. This is the genetics. And when we originally found the mutation and realized that it had a pretty strong colon phenotype compared to the mouse, we wanted to know, all right, what are the genetic control of that process? So we did a number of different crosses. We crossed it to ACI, we created that F1, and you can see that there's an obvious modifier that enhances that. This is all in male. There's a pretty stark difference between male and female, so there is a sex effect, and I'll talk a little bit about that later. We then made a congenic and ACI, and you can see that those tumor numbers are about double of what we see in the Fisher, and that's actually even at an earlier age. And then Brown Norway is kind of the opposite, right? And the Brown Norway dramatically suppressive effect, and some of those animals don't get disease at all out to 6 months, almost a year. We don't see tumors in some of those animals. So there is clearly a genetic effect here, and to resolve that, not really sure how to approach that in an economic way, but we're trying to figure that out. So one of the things that I'm interested in is not only kind of the variation in human genetics, but we wanted to make sure we were controlling the environmental conditions. And this is mouse data. So as part of the Mutant Mouse Resource and Research Center, Craig Franklin, who was the director at the time, and Aaron Erickson and several other postdocs, were asking the question, we're getting mice from all around the world. What does the microbiome look like in these mice? And this is representative. So what you see here is stacked bar charts. Each one of those colors is the representative amount of a different bacterial, we'll call it a species, sometimes it's order, sometimes other phyla level. But you can see that pretty much every mouse is different, and it really varies across the spectrum. And this is at a fairly high level. But you can see we have very rare taxa or animals that have very few bacterial species and some that have a ton. And the big question is does that have an effect on phenotype? And we've done a lot of mouse studies related to this, looking at everything from autism and behavior to the colon cancer syndrome to, and we did the APC min mice, as well as DSS treated mice. And microbiome has an effect, and it's really pretty dramatic most of the time. And it's something that we aren't really thinking about. So if you walk away from anything from this talk, it's, okay, maybe I should probably be putting some poop in the freezer, right? At least know what I have at some point in time. I don't have to do it now, but if things change, I should probably make sure that I know what it is. So the way we asked that question in the rat is we took embryos from the Fisher 344 PERK animals, transferred those embryos into different surrogate dams that each had different microbiomes. And these are just off-the-shelf SPF, they don't carry anything that we would consider, that we would want to eliminate from our colonies, and then just followed that over time and said, okay, if we change the microbiome, what happens? And in fact, it changes quite dramatically. And so this is what we had published previously. And you can see that here, these are just colon tumor numbers. The most remarkable thing in this is this animal right here. This is a male on here that got no tumors. This was the first time we had ever seen the phenotype actually go away. Okay, I told you that genetically we can do that with Brown Norway, that it seems like there's strong suppressive effect. This was the first time we saw it. And we actually saw it in a female as well. This is just the male data. And so it sparked the question, what's driving that? And so now I was at a meeting and I told somebody I was working on the microbiome because, we were trying to figure out the genetics, but we wanted to control the microbiome. And he said, that's so complicated, you'll never be able to figure that out. And so when I saw this graph of what are the different species that are controlling that process, it is incredibly complex, right? So how do you go after this? And this became kind of the complex question. You have these really complex systems with thousands of different microbes in there, and they're all at different varying levels. And you know that they're driving the system. You know that you have an effect due to this, but how do you know who's really the player there? And is it a single player? Is it multiple players? And so we wanted to take a reductionist approach. Currently you can't get commercially available germ-free animals. So we were like, all right, what's the most limited microbiome we could get? And so we reached out to Charles River. So Charles River, and this is information that maybe all of you don't know this, and I got this from Charles River after some conversations with them, how they refresh their genetics. They obviously have colonies all over the world where they distribute their models. Every 5 years, they actually bring all of those back to their main facility and they intergress the genetics. So they breed them all together, they create embryos, and then they rederive them onto this Charles River altered Shadler flora. It's a very limited microflora of 8 microbes, I think. Yeah, it's about 8. And that's what they start with. That's their nucleus colony. And then they put them into their normal, barrier centers, and then they get populated with with whatever's there. So Charles River that you get from Charles River out west is going to be different than out east. That's going to be different than Asia because these ASF rats are sent to those locations. And so we asked the question, hey, can we get some of these, like the ASF ones? And I told my postdoc, or my student at the time, I said, it never hurts to ask, just ask him, can we do this? And of course they said no, because they don't do that. They don't sell those animals. Those are the ones that they use for expansion, and they're very limited. So I said, well, you can ask again. I think we asked a year later and said, just ask. And they're like, well, actually, yeah, I guess we could probably sell you a few of those. We could just ship you some and ask that question. So we received the Lewis. I was unfortunate. So the year before, they were actually rederiving their Fisher, which was the genetic background that we had the original line on. Unfortunately, this year they were doing Lewis, and that's all they had available. So we were like, all right, well, we've never looked at Lewis F1, so let's see what we can do. And so we maintain these on an IVC rack. So we get them in a germ-free kind of, well, not a germ-free, but an isolator, a notobiotic isolator with these rats. And we put them on the rack. So these on the left here, this is the same microbiome profile. You can see most of it is Parabacteroides, this light blue. And that's what we got. They sent us poop before they shipped them. And then we looked at them after they were on our IVCs for a couple of weeks. And you can see it's pretty close. They actually did pick up a few. These brown blocks here are not over here on the left. And we went back and looked at things that we sampled in the isolator, and it turns out that we picked it up from their gel packs that they used. So those are not sterilized. I think they're irradiated, but they probably still had spores in them. So we found the same bacteria in the gel packs when upon arrival. So essentially did the same experiment. So I want to point out here though, to get the animals that we had onto this microbiome, we used rederivation. We took the uterus out, dipped it in betadine, took it into the unit under a hood, and actually fostered a pup. We actually fostered a lot, but only got one, happened to be a male. So we were really lucky. So this is the fostered male at about weaning age for him. And he looks a lot like the dams, so pretty limited microbiome. So we use that male to mate to the other Crass-F animals, these Charles River altered Shadler flora rats, and then created F1 hybrids. And then at weaning, we separated those. We said, okay, we're going to keep some in this barrier status, just Charles River altered Shadler, just to see what the phenotype is. And then we're going to take some and we're going to co-house them with other conventional animals. They'll pick up a microbiome from those conventional animals, and we'll see the difference. And so this is at weaning. So everybody looked about the same. These were the dams here. This is the sire. These are the ones we kept in barrier. These are the ones that we moved to conventional before we moved them there. They look all pretty much the same, only about 11 species, so pretty good. This is what happened after conventional at 4 months. So clearly they pick up the other microbes, which is great. That's kind of what we wanted. And these actually maintained a pretty good status, although you'll see that the levels of those bacteria change, and we certainly know that happens. Clearly they are different. We see that they have a lot more species, a lot more OTUs, as we call them, or ASVs. And you can see that the diversity indices go up. So what was the phenotype? It kind of caught us off guard. It turns out barrier animals actually had more tumors in the colon than the conventional animals. And that totally, I was sitting there, and of course, I feel like every student project that I've had, we go in with a prediction and it's always the opposite. Which really disappoints students, but I'm like, no, that's even more exciting because now there's only one of 8 species that's driving up tumorigenesis. So it might actually be a really cool finding. And this is in the small intestine. Now I told you this was largely a colonic phenotype. I didn't show you the data. They do get a few small intestines, but it's really relatively rare in our normal colony in fishers. This was interesting. These are the small intestinal tumor numbers. And it's the exact opposite. So really what we've done is we've shifted from a colonic phenotype to more of a small intestinal phenotype, or at least about equal. And you can see that there's a sex effect here as well. So there's something about those bacterial species where they live, what metabolites they're potentially producing that is now shifting that phenotype. So I had an undergraduate in the lab. I said, well, you know, why don't we look at the intestinal epithelium, see what's maybe driving this by looking at some of the cancer-related genes. She just did some quantitative PCR this past summer. And it looks like that there's an increase in DNA repair pathway, the MGMT. So there's probably DNA damage going on. The CYP27A1 is a bile acid producing gene that's mostly expressed in the liver, but obviously expressed in the intestine as well. And it looks like that's going up, and probably in response to the different bacteria that are driving that bile acid production or secondary bile acid metabolism. So it's an interesting prospect, and this is kind of where this project sits. And it's always been kind of this back burner project that we really would love to see move on. So it's kind of interesting. But it brings up a bigger topic, right? What should you do, right? So first of all, ask, could this matter? And anybody in this room, I imagine it matters. Behavior, it's really a big driver of behavior, but was actually exceptional. If you really want to see a video, I have a picture of mice. They're outbred mice on 2 different microbiomes, and the marble-bearing phenotype is so dramatic between those two. So if you want to see that, I can show you that. And then think about what is your phenotype, right? If it's quantitative, I guarantee it's going to have an effect. And so what can you do? Think about it. If you're receiving animals and your unit requires you to rederive your animals to get them into your institution, ask where they're getting their surrogate dams. If you want it to be similar to what's in your colony, see if they'll use some of your colony animals as surrogates so that you have the same microbiome in your colony and that you're not introducing anything new. It's pretty cheap to do a metagenome profile. So, you know, if you don't have money to do it now and you want to know if it changes when I move to one institution, just throw something in the freezer. So be proactive about it and really ask those questions. And we can help you. This is the MUMC. If you want to scan that, we can do screening. It's not that expensive to do that. So we can sequence your poop for you and give you a profile. This is the group. I conned all of the Rat Center people to put Mouse Center shirts on. So we actually share a lot of the same people. So it's great. So you can see Elizabeth in here. I'm going to highlight Craig Franklin, who's really the guy who he's now an emeritus professor, but really the brainchild behind really looking at the microbiome as a driver of phenotype. And this is our poop group down here on the left. This is an old photo, but these are the kind of the people that did it. Sushil here is now over in the UK and is doing microbiome profiling in soil and environmental samples. And Jake here that I've highlighted is the one who did the mouse work. So with that, I'll take any questions.
Not a question, but it's a statement. If you or anybody else here wants to make true notobiotic rats, paleontology medicine is capable of doing it. They imported from France the only clean notobiotic rat colony in existence that I'm aware of.
So when you say notobiotic, do you mean germ-free or is it
germ-free?
Oh, that's good to know.
They do germ-free mouse colonies. They can do rats. They don't. So they maintain the foster mom colony, but they're not currently doing it. I've been trying to get it to do some point.
Yeah.
So it's only a matter of.
Yeah, yeah, I know. It's all about the money. Yeah.
I don't have a microphone.
Oh yeah, sorry, I can repeat questions. So Baylor has the notobiotic rats if you really want to do germ-free rat experiments.
Hi, can you talk a little bit about going for housing?
Yeah.
I don't know if you have different phenotypes, and also like the number of non-specific that are housed with. Did you see any effects in your research, and how do you control that?
Yeah, so we've published on some of the housing effects of these guys. So we pair house usually at least, and kind of maintain them. I'm trying to think of other questions. So autoclave feed or at least sterile feed, we use a radiated feed to keep ours, and that's a few. And then thing has been a I don't know, it's been a problem, not so a problem. We used to have paper chip bedding, which we loved. It was very low odor. It was really nice absorbent material. They no longer make it, or at least we can't get it anymore. So we've moved to corn cob bedding. I don't like that as a bedding, but I don't know. What are those the kind of questions you might have? It's usually always at least 2 or 3 females sometimes. And the microbiomes definitely will be variable between the two, but it's, you know, you'll see the same species just at relative abundance differences. And that varies. I mean, that can vary through the time of day. You know, you try and be consistent when you take samples. So all of those things.
Okay, thank you.
I'm gonna take whichever one you want.
Okay. I'll try to start quickly because I think we're running a bit late. Well, I'm really excited to present to you today. And so this is a story that I've been working on for a long time. It's finally coming together, and I'm at a point where I'm really looking for feedback and also potential collaboration. So please talk to me later if you can. So, yes, okay. So first of all, I wanted to, can you hear well? Yeah. Yes, so I wanted to take just a moment to motivate why we do this microbiome work in outbred laboratory rats. So first of all, you might know that from the news, for example, that in humans there are tons of correlations, associations, but really correlations that have been reported between the microbiome and pretty much everything. Obviously, a very big question is whether these correlations reflect causal effects between host and microbiome or just confounding from environmental factors. On the other hand, there's been a ton of experiments on the microbiome in the lab, and these experiments, for example, have used germ-free animals or mouse knockout models, genetic knockout models, which are all pretty extreme interventions, I would say. And these experiments have in this way shown that the microbiome can influence the health of the host. But there is, I think, to me, a question of whether the microbiome actually influences the host in more normal conditions in the absence of such extreme treatments or group comparisons. And so I want to argue that studying host microbiome interactions in HS rats is actually combining a bit the best of both worlds. So on the one hand, we do, first of all, a lab experiment, so we control the environment quite a lot. We also can design the study in such a way that, you know, we limit confounding between the things that we care about. All the variation that we look at in terms of genetics come from natural variants. They're not engineered variants. They are variants that are actually segregate in wild populations, and I'll show you an example of that today. We use rats with a normal microbiome. I personally don't really like germ-free animals, and we don't treat them with antibiotics either. So it's just like regular rats, pretty much. And then we can use host genetic effects to determine causality using mediation analysis, for example. This is very annoying. I don't know if you can you still hear like this? Good enough? So today I'll show results that come from an analysis of over 4,000 HS rats. We don't need to introduce the rats anymore, but of course, this is not something that I did. This is something that was done in collaboration with many, many people. So you've heard many times now about the NIDA Center. I wanted to point out here some of the PIs who are involved in the particular data that I used. But of course, all their groups also should be thanked. So I'm really grateful for all the work done and the great collaborations, actually. And for the microbiome park, this was done in collaboration with Rob Knight at UCSD, and that's actually a project that was initiated by Oksana here and Abe many years ago. And it's taken me many years to analyze the data, so thank you for your patience. So I'm looking at over 4,000 rats here, but they are spread across 4 cohorts, which match the P50 cohorts, right? So one comes from New York, one from Michigan, 2 from Tennessee. In Tennessee, we have breeders and we have adolescent rats whose behavior was phenotyped. So these cohorts, really, they differ in many different ways. I'll just point out a very few here. So of course, the facility is important, as we've just heard. The rats pick up the bugs that they are exposed to in the different facilities. So that's an important difference. Of course, there's also differences. Well, there's also an age difference, which we think is important for the microbiome. There is evidence that age is important. And fasting, in our case, was also different. So some of the cohorts were fasted overnight before the Zika were collected, and one was not. And there's also differences in sample sizes here, which do affect the results, as you'll see. So different cohorts, and as a result, we see overall different microbiomes. So this is kind of a PCA plot showing you the overall composition of the microbiome. What you can see here is that you have 4 different clusters corresponding to the 4 cohorts that I just presented, right? And so as far as I'm concerned, I'm just studying host microbiome interactions in 4 different cohorts that vary and that are representative, I think, of the variation you can expect and you necessarily have in microbiome studies. And I decided to analyze those cohorts separately. And I did this because my 2 key questions were the following. So the first one is, to what extent do host genetic effects depend on the environment? So to what extent do we see different things when we look at these different cohorts? And also, of course, can we identify host genetic effects that are robust, that are replicated across the different cohorts? Because if we're going to do any follow-up on the things that we find here, it's quite likely that it's going to be done in a different facility or perhaps in the same facility, I don't know, but maybe different bugs. So we only want to work with effects that I think are robust enough that we can expect to see them in another facility. So first of all, I looked at the aggregate level. So I'm talking about heritability as opposed to GWAS, the sum of genetic effects over the genome. And because I think my talk is a bit long, I'll just sort of give you the conclusion of this. The conclusion here is that heritability in one cohort is not predictive of heritability in another cohort. So if you have a bacteria that's measured in one cohort, say in one facility, and we find that it's really heritable, affected by host genetic effects, it might not be in a different cohort, okay? Now, a separate question is, assuming that we're looking at a bacteria that is heritable in 2 cohorts, for example, in Michigan and New York, as we see here, is it the same genes or genetic variants that affect the abundance of this bacteria, and is it also the same effect sizes, right? So let's say if we do the idea is that's a genetic correlation I'm talking about here. So if we do, for example, if we did the GWAS in one cohort, in the other cohort, and we correlate the effect sizes across all the variants, what correlation do we get? And we do get correlations that are above 0.5 on average. And I was, scrutinizing the figures that we saw yesterday to figure out whether this is actually a high correlation or not. And I do believe that it's a correlation that's actually pretty high, although I think we might need to do a bit more work to contextualize this and see whether body weight measured in one cohort is very highly correlated with body weight in another cohort too. But anyway, clearly genetic effects are shared to some extent across the cohorts. So then can we identify genetic effects that really are shared across cohorts? I don't know how it comes out on the screen, not very strong here. But we did identify 3 QTLs, let's say, that were replicated in 3 or in some cases in 4 cohorts. So these are QTLs, and you can see, so first of all, so chromosome 1, 4 and 10, right? You see these peaks, they're super big. So they are very significant, extremely significant. And in all 3 cases, evidence shows that they are replicated across multiple cohorts. So just to give you something to compare to, in humans, there's a single locus that really is replicated across populations, across cohorts of the same populations usually. It's the lactase persistence locus. And so here we did identify 3 of them. It's really promising, I think, and no doubt, it's also because we are limiting environmental variation in our study. So I have sort of a nice biology story, I think, that I want to get into that, and it's about the 3rd QTL here on chromosome 10, and it's a QTL that affects the abundance of a bacteria called Paraprobotella. So as usual with QTLs, our questions are, what are the causal variants or variants? What are the causal genes? And what's the underlying mechanisms? So first of all, and this is done, this is work that was done by Dan, who's over here from the Palma Lab. So Dan looked at long-read sequencing of the 8 HS founders, and he found that there's a partial duplication / triplication of the ST6-GalNAc1 gene in the WN founder of the HS. So what you can see at the bottom here on the top track is the depth, the sequencing depth across the genome for the WN founder, and you see these steps here that really clearly show a duplication, and then part of it is even triplicated. And in fact, in that region, RGD told us that WN and SHR are identical by descent. So thanks to Peter Doris, we have a de novo assembly of the SHR genome. And when we compare the de novo, well, the assembly of the SHR genome with the assembly of the BN genome, we can clearly see the duplication and triplication that I showed before, right? So we have 2 lines of evidence. It's very clear that both in WN, founder of the HS rats, and in SHR, there's a copy number variant here. So we have, well, okay, so let me tell you this as well. So this locus actually was reported previously to be a locus under selection. It was reported as a selective sweep. Perfect. And so basically we rerun some of the analyses that were reported in the papers. So this is something that Jorge, a collaborator of mine, did. So he used publicly available data from wild rats, some of which were collected in China, some others in New York City. And then he also, put on the same map here inbred lab strains. So what you're seeing are the animals in rows and the genotypes in columns for a region surrounding the ST6-GalNAc1 gene. Okay, we have pink, blue, and blue for the different genotypes. And so what we can see here, first of all, is that and actually that's, I mean, I don't know that this slide specifically shows that, but the exact same duplication, triplication is seen in the wild rats. Okay, so the variant we're looking at segregates in the wild, first of all. And what you can see here is that at the locus, there's only 2 haplotypes. It's either pink or it's light blue. And actually the light blue, it is not really het, it's homozygous, but because there's a duplication, triplication, and there are differences, it shows up as het. There's only 2 haplotypes that are segregating here, whereas you can see around, it's very random, right? It's very patchy. And this is the signal that was picked up to say that it was under strong selection. So the fact that it's under strong selection does tell us that the locus is functionally really important. It doesn't tell us how, but it does show that the variant that we're studying is evolutionary important, which is really important to me. So something I wanted to point out here, just because of the audience that is here, is that there's a similar copy of that gene in mice, actually. It was reported a very long time ago, and having genetic variants that are conserved between 2 species like mice and rats might point to balancing selection, long-term balancing selection, which is very interesting. If anyone sort of has anything to say about it, please come and see me. And this is actually a variant that in mice has been studied for its consequences on type 1 diabetes in non-mice. So again, anyone wants to talk about it, let me know. So getting into the mechanisms now, I'm showing you here all the bacteria whose abundance is affected by the locus and Paraprovotella I've already told you about. Now, if we go at a level that's below the threshold, we see that Akkermansia is also affected by the variant. And that's a very well-known bacteria of the gut. It's very well known because it feeds on the mucins of the gut. The mucins are proteins that are heavily glycosylated that line up the gut epithelium, and they are very, very important for gut health and host microbiome interactions. And the short story here, again, for time, is that it looks like the effect of ST6-GalNAc1 copy number variant on our bacteria is likely mediated by the sialylation of mucin. So ST6-GalNAc1 is a sialyl transferase, and it likely sialylates the mucins and affects it should go ST6-GalNAc1 sialylation and then bacteria. All right, so this is what I just told you, variants to bacteria, and we kind of guessed the mechanism. Now, are there any consequences on the host? Well, we and I, when I say we, this is not we, this is the work of Tiago again in the Palma lab. Tiago is sitting here. He did a PWAS with all the HS phenotypes, and none of the phenotypes collected in the HS were actually affected by this variant here. Now, there are things that are known about Paraprobotella, and it is known to prevent IgA degradation by trypsin in the gut, and that has consequences on infection vaccines. So I think we, if we want to follow up on this, this is the direction we should go. Then Akkermansia, but also Paraprobotella are both associated with IgA nephropathy, and I think due to what is known about Akkermansia, we have a potential mechanism here, so that's another thing I'd like to explore. And of course, the mucus cellulation is known to be really important, as I said, for the gut. And if you compare the gut barrier function and response to infection, you see better or worse. This is saying that on one hand, it's good, on the other hand, it's bad. And that could be something underlying long-term balancing selection. So that's a really interesting thing to follow up on. Okay, I'm going to wrap up. I'm looking at gut microbiome transmission, but I think I'm over time, so I'll skip this. I just wanted to give you a sort of an overview of the rat microbiome projects going on in the lab. So thanks to Michal Pravenek, we recently got fasted rat Zika from the BXH panel, 30 strains and replicates within each. And so I'm going to see if the association we see with Paraprobotella is replicated in this entirely different population. That would be very, very interesting. Where I put in red here the sort of resources that would be useful for the community. So we're generating a lot of shotgun sequencing of the microbiome. And we are going to have a catalog of bacterial genomes for the rats, because this is really underrepresented in current databases. So I think this is going to be useful for anyone doing microbiome work. Work to do with the metabolome as well. At the moment, what is coming up is a paper from AIDS Lab and led by Joel about metabolome GWAS. Okay. So please think about putting some poop in the freezer. That's the word, okay? And if you're interested to collaborate, let me know. And I want to finish by thanking, so first and foremost, the P50 collaborators, of course, and Abe, because he was really, really important for me to get into this collaboration. People in my group who have worked on the microbiome and on the code, Felipe and Hélène. So people not only in Abe's group, but also in the Rob Knight group at UCSD, Jorge, who's in Barcelona, and the RGD team for all the help, especially with the variant visualizer, which is like the one most by far. So that is really, really cool and funding. Thank you.
No, it's actually going on. Oh, microphone working.
Maybe just speak out, because we have to repeat the question.
Well, we have a long rally. Thank you very much for demonstrating the diversity. I talked a little bit about it yesterday in the immunoglobulin lupus. The most abundant immunoglobulin protein of all these is IgA. We make it secrete into our gut where it's sort of farming material to help maintain the microbiome. And as I showed you yesterday, there's enormous genetic variation. Yes, one of the reasons it's evolved is to really help maintain the microbiome. what we tend to see is that the expression of immunoglobulin gene repertoires in the gut is much more limited, particularly on common molecules that are like pathogen-associated molecules. The first comment. Second is that I'm always worried when doing microbiome studies that we don't have sufficient resolution on many times to do deeper sequencing of the organisms ourselves. I'm only reminded that you can go to McDonald's and buy the wrong hamburger and you get an Escherichia coli that will kill you, whereas most of your gut's lined with Escherichia coli all the time. And so some things are not necessarily the same between different colonies. Well, maybe the variants of the organisms are not the same either. What do you think about that?
Well, so I think when we think about the microbiome, very often we want to get beyond the taxonomic definition of the bacteria. What matters is really the functional, they're not only metabolic, but more generally immunogenic profiles and so on. So the function of the bacteria, and there's nice work that shows that, for example, between mice and humans, there's different bacteria actually present in the gut, but at the functional level, there's a lot more sort of overlap, of course. So yeah, we definitely want to look at the bacterial genes. So we need the genome sequences of the bacteria that we are seeing, and then we'll look at the genes, the function, and that's how we want to go ahead.
Thank you.
Thank you.
That was very interesting. He's finished there and perfect. He did, he already did.
All right.
And we'll bring his face up. If he's on the screen. Oh yeah, I see it, Evan.
I'm there, yeah.
Yep, excellent. We're switching the visual. Not me. Can you say a couple words, Evan?
Hi, I'm here.
There we go.
All right,
Keep
great.
talking.
Yeah.
Okay, so you just saw Alan's face a second ago if you didn't know him. So I guess I'm staying here because next year I am proposing to organize the CTC-RG Genomics meeting in Barcelona, in my hometown, well, work town. And we have Evan here who has volunteered to help me with that. Piotr has also volunteered to help. Actually, you know, if anyone wants to join the team, just come and see me. That would be great.
More than one is good.
So, I mean, I guess the first question is, are we doing it in Barcelona? Is there any other
show of hands?
I mean, was there anyone else who really wanted to organize next year? So, okay, let's take that for granted. It's going to be in Barcelona next year. We have worked quite a lot already on the dates to find dates that would not clash with, you know, the other meetings and various commitments of the different groups attending. It looks like the best one, and it's really actually quite hard to find those dates, is 21st, 24th of October. Please think about that, okay? And if you realize there is a clash, I would love if you could drop me an email to let me know about that, and we'll see what we can do. For now, we have already pre-booked the auditorium. So my research institute is in a building that has also many other research institutes and a university. We have a big auditorium, and what you can see here is just the outside of the auditorium. We're just by the sea, so it's really nice. So we can have, you know, snacks over there, poster sessions, and so on and so forth. We're also really in the center of Barcelona. So we're not saying here, you know, conferences or holidays, but certainly since it's going to be Wednesday to Friday, if you want to extend a bit, you will have a chance to enjoy Barcelona. And actually also, you know, it's going to be nice to go to the restaurants. There are tons of restaurants around and so on. So it should be fairly logistically, I think, quite simple. Oh, I had a paper with things I want to keep.
Is this paper?
Yeah.
Yeah.
So we'll talk about these. Oh yeah, so one thing that seems to be really important to everyone here is that trainees get talks at the CTC. Okay, and so we'll keep that. I think it's very nice for trainees to have a chance to present. The exact format we'll still, sort of discuss with the team and so on. We were thinking, and actually Piotr suggested this this morning, just after waking up, to have workshops organized by outside people, perhaps. Not the kind of workshop organized by us, but maybe a workshop on Nextflow, for example. I don't know if you're familiar. Nextflow is a tool that's really good for developing pipeline, and it comes from the CRG, my institute. So we could get like a training by someone professional at Nextflow and other tools. Evan also reminded me that it would be nice to have career development a session, like mentoring session, which we'll try to do as well. So Evan has been working with Arthur, also in Rod's lab, on the website for next year's CTC, and it's almost ready. So I think it will come up shortly after this meeting. And finally, if anyone has tips on how to talk to sponsors and sponsors we could talk to, that's really important. I'm hoping to go for a registration-free meeting, but that means we need to get a few sponsors to be able to, pay for coffee breaks and ideally lunch as well.
Talk to Mary Kaldunsky. She's our expert.
Super. Let's do that. Okay, I hand over to Evan. Evan, did you want to add something?
Oh, you're muted.
No, not much else for me to add. Just we'll organize and get more details out to you, but just wanted to make sure the dates so we could start advertising and getting people in for next year in Barcelona.
Good. All right. Thank you.
Thank you for organizing this.
We have lunch right now, and it's again over there. And right after lunch, we're going to do the poster session, which is why the fact that we ran a bit over, we can make up the time. So get your lunch, have it, and then go to the posters. And we will be doing that until 2:15. So you do have a little bit of time. I don't know if it's as warm outside as it was yesterday, but you can give it a try. We'll prop the door open. So enjoy your lunch. Anybody who needs gluten-free, there are packages of buns next to the other buns. So if you don't necessarily have to have gluten-free, please don't take those. Thank you.